books, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Has studied over the course of your career?
Sure. So my name is Joe Takahashi. I'm at UT Southwestern Medical Center in Dallas, Texas. And my lab is really known for discovering the first circadian clock genes in mammals. And so the first gene, which is actually named clock, because back then we could name our genes when we first discovered them, turned out to be a transcription factor, works with another protein, and it turns out to be the master transcription factors of the circadian clock transcriptional gene network, which is what actually makes up the circadian clock. We can get into the details of that if you'd like.
Yeah, I think I do want to get into the details of the transcription translation dynamics and sort of the nuts and bolts within cells eventually. But first, I want to just get people oriented at a high level to, the idea of circadian rhythms and sort of the phenomenology or ecology of circadian rhythms. So before we get into the nuts and bolts and the mechanistic stuff, at a high level, what are circadian rhythms first? And why do animals and organisms need to entrain themselves to the rhythms of the physical world?
Yeah, sure. So, obviously we live on a planet that has a 24-hour day. It's driven by the rotation of the Earth, which generates a 24-hour solar cycle. And if you were a plant, you would say that this is an energetic cycle. You harvest energy from sunlight in the daytime, and then you might do other things at night in the dark. And so we actually kind of look at the environment actually as an energy cycle also. Now, we're not plants, obviously, but our bodies go through energy cycles every day. We go through an oxidation and reductive phase every 24 hours. Our entire metabolism fluctuates on a 24-hour basis, and it's optimized really to deal with our behavior, which is also diurnal or varies with 24-hour rhythms. Now, obviously, our sleep-wake is a major controller of our behavior, and that partitions our behavior into, a rest phase and active phase. And we do many different things in our active phase as compared to our rest phase when we're sleeping. Now, to sort of step back from our own clocks, because of this environmental cycle that life on our planet evolved under, what happened is that over time, organisms evolved clocks to really anticipate the time of day and this energy cycle. And so plants in particular are extremely tuned to the 24-hour cycle. And the majority of their genome is actually regulated by the circadian clock in order to take advantage of how light and dark change and how they photosynthesize, and then store that energy over time. And so, uh, because of this pervasive 24-hour cycle, uh, almost every living organism on the planet that's at least that is exposed to the light dark cycle uh, has evolved and uses a 24-hour clock that's internal, uh, really to anticipate these changes.
Mhm. So, like one question that comes up here is if we're on this rock floating through space and it's constantly going around the sun, and that is like truly at one level a constant in the environment. Obviously, you've got cloud cover and things like that that change, but if it's a constant, why does the system need to be able to anticipate and build expectations around when things happen? Does this perhaps have to do with like a lag time it takes to boot up or boot down certain processes that need to already be in place, say when the sun comes out? Like naturally, I would think a plant needs to get all of its photosynthetic machinery, whatever exactly that means in total, in place to fully take advantage of the sun. And in order to do that, maybe it needs to get that stuff ready before the peak sun it's going to generate its food from is actually out. Is that one way to start thinking about why this is an anticipatory and modifiable type of thing?
Yeah, that's one of the major reasons is that many processes that, would function optimally at a particular time of day can't be just switched on immediately.
Mm-hmm.
They require perhaps synthesis and building of certain pathways that take perhaps hours each day to really achieve efficiently.
Hmm.
And so, we can't be completely reactive in all of our responses. I mean, you could imagine the plant could just, react to when the lights come on. But uh, I think it's clear that that's less efficient. And also, the time constants are very long.
Right, right.
Um, the other issue in the environment, as you mentioned with cloud cover, is that you don't want to be responding on a short-term basis to how intense the light is
Right,
uh,
right.
throughout the entire day. We're just sort of integrating the light information
Mm-hmm.
over long durations, and it's just a much more stable system that way.
Yeah, yeah. And I think hopefully, if we do a good job, people's intuition for this will kind of become stronger once we dig into some of the details around transcription and cellular level processes. But, by way of analogy, like, I'm on a MacBook Pro here to do this podcast. When I wake up each morning, I come into my office, it takes seconds or a couple minutes for it to boot up. And that's really, really fast. And we probably all have an intuition that, you need to boot up and boot down systems, both, in our lives. We know that, it takes us a few minutes to get excited or to become restful at the end of the day and things like this. My computer takes a couple minutes to kind of boot up and for everything to load before I start actually using it. And the boot up and the boot down in biological systems, maybe we can think about them as needing more time because, your synapses operate at the scale of milliseconds to seconds. But when we start thinking about genes turning on and off, trafficking proteins, repairing damage, now we're talking about things that take minutes or hours of time. And I think all of that kind of relates to what you were saying. You can't just respond immediately to the environment because the time constants, the timing of biology just requires more than that.
That's right. No, exactly.
And so when we think about, I think we're going to talk about the whole organism, but we'll probably spend a lot of time up front talking about the brain and what some people call the core or central clock of the nervous system. So let's just start very basic. What is that and where is that in the central nervous system?
Yeah, so in the early 70s, it was discovered that there's a nucleus, a cluster of neurons in the base of the hypothalamus. Uh, that it was discovered because it received a novel projection from the eye, which is called the retino-hypothalamic tract. And so um, the people who discovered that thought that they could interrupt that visual input by lesioning the terminal nucleus in the hypothalamus, which is called the suprachiasmatic nucleus. Supra for above the optic chiasm. And there are only about 10,000 neurons in each side in the nucleus. So it's not a large structure.
In humans, you mean?
No, I'm talking mainly about rodents.
I see. I see.
In humans, it's actually not very big either.
Bigger than that, but not that big.
Yes. Relative to the human brain, it's minuscule, I would say. So the surprise happened that when they lesion that nucleus, instead of interrupting the light input to the clock, which would, they thought, disrupt synchronization of, say, rats to a light-dark cycle, it abolished the rhythm. In one case, it was a drinking rhythm. In the other case, it was an adrenal rhythm. And so that discovery was made in 1972 by Robert Moore, his lab, and then by Frederick Stephan and Irving Zucker independently. And so that was really an interesting result back then because this structure in the brain of unknown function had such a major impact on the behavior of rats. And so, many people followed up on that work and showed that, you could lesion the SCN in many other rodents, the hamster, later mice, and you'd find the same effect. And so back then it was hard to study circadian rhythms because it was either a behavioral endpoint in a rodent, but to study the brain structure, their early attempts used brain slices and then recorded electrophysiologically from the suprachiasmatic nucleus. And what they found is that SCN neurons have a spontaneous circadian rhythm. They fire in the daytime, and then at night in rodents, they go completely silent. But it was very difficult to do that kind of experiment. The slice, it was hard to keep the brain slice going for more than 24 hours, and no one actually could record continuously for 24 hours.
Right. So you could record from a slice, but of course it's divorced from the actual brain, and so you always wonder in that case, well, is it now behaving the same or sort of the same or not? Right. But you're in essence, they were able to record at this time from a narrow, like a time slice, and they could look at these neurons and be like, okay, for this time point in the day, this is how the neurons are firing. And then at a separate experiment, they can take a slice at a separate time. This is how the neurons are firing. And they started to discern, it seems like the neurons in this part of the hypothalamus are firing or not, mostly based on whether it's light or dark. And therefore, if you sort of stitch that together, the idea was, okay, maybe they fire in this kind of circadian daily rhythm.
That's right. But I would say progress was difficult because the measurement of the rhythm of the SCN was just technically challenging back then. So, if we fast forward to more modern times, then in around 25 years ago, I guess, no, I'm jumping ahead too much because that required the discovery of clock genes. Which then enabled us to make reporter mice that used a circadian gene that cycled, driving a reporter such as firefly luciferase. And then we could follow luciferase in an SCN explant, and a lot of technical advances were made so that those slices could be cultured for many days to weeks.
So technology advanced over time, getting you to a position eventually that you could watch, literally see and measure when genes were active or inactive in these neurons for extended periods of time rather than just studying them for, an hour at a time.
Exactly. Right.
And so as that technology progressed and you could look at neurons, you could look at genes, a lot of your work is happening sort of inside the cell. What did you guys start to piece together once the technology was at that level?
Yeah. So I think I jumped too far ahead because we sort of skipped the gene discovery phase, which was really the second major discovery in the field of circadian rhythms. The SCN, was discovered in 1972, but in 1971, the very first mutants that affected circadian rhythms at the behavioral level were discovered in the fruit fly Drosophila
Mm.
by Ron Konopka and Seymour Benzer. And so Ron Konopka was a graduate student with Seymour at Caltech, and he searched for mutants. He did a mutagenesis screen, making random mutations, uh, and then he found three lines of flies that had altered circadian rhythms. So one of them had no rhythm. The other had a short period clock, about 18 or 19 hours, and the other had a long period clock, about
Hmm.
28 hours.
Hmm.
And he mapped those three alleles to the same locus, suggesting that they were the products of one gene.
Hmm.
And many decades later, we know that's true.
I see. So it was three different types of mutations all in the same gene.
Yes.
And
So,
yeah, go ahead. Go ahead.
yeah, incredibly lucky that his first three mutants were in the same gene. Now, part of that was the way they did the screen. It was focused on the X chromosome only. Um, so it sort of narrowed the scope of the screen, but still there could be other genes, I would
Hmm.
imagine.
Hmm. Hmm. And so what was this gene? What did it end up being, and what does it do?
Yeah, so they named it the period locus, and today it's the period gene. Uh, that gene wasn't cloned until 1984.
Hmm. So we knew about this general region of that chromosome, and it clearly had something to do with these circadian rhythms. And then it took a little
Mhm.
bit more time before we said, oh, it actually is just one gene in this chunk of chromosome.
Yeah, so from the original work that Ron Konopka and Benzer did, their evidence was pretty good that it was a single gene
Hmm.
because they could find map the location of those mutants, and they mapped to a very narrow region. Um, so it looked like it was one gene even at the very beginning.
Hmm. And so
Yeah,
and so
so
walk us
that's a
through.
classic paper. It's a beautiful study.
Yeah, yeah, yeah. And Benzer, for those that don't know, Seymour Benzer is one of the more famous and admired scientists in the general realm of genetics of all time, really. And okay, so what happened when we started to get to the functional stuff and figure out, like, okay, what does this gene encode for, and what is it doing that gives it this connection to these rhythms?
Yeah, so once the gene was cloned, the predicted amino acid sequence uh, was not very helpful because it had no obvious protein domains that
Hmm.
would give you a clue to the function of the period protein. And so there were a couple red herrings, early on. People thought it was a cell surface protein. They thought it was a glycoprotein. Um, those initial um, suggestions turned out to be incorrect. And it wasn't until many years later they discovered it was really a nuclear protein primarily expressed in the nucleus. And that eventually led to the idea that it might be a transcriptional regulator, even though it didn't look like a transcription factor. In fact, it still doesn't today.
So the sequence, when you look at the genetic code of it, it doesn't really scream transcription factor, nuclear protein, but it turns out that that's what we're talking about.
Yeah, it's not a transcription factor, strictly speaking.
Mm-hmm.
It's a transcriptional repressor.
Mm-hmm. And so this might be a good spot to start like defining some of these terms for people. So when we talk about proteins that affect gene expression, things like repressors, things like transcription factors, what are these things? How do they relate to the turning on and off of genes?
Right. So transcription factors can be activators or repressors, but what makes them different from, say, just a repressor molecule is that they actually bind to specific sequences in DNA. So they have a particular binding site motif. And so transcription factor binds DNA, and then it works usually in concert with other cofactors to either activate or repress transcription.
Mm-hmm.
Okay, so in the case of the period protein, it doesn't bind DNA directly. So that's why I said it's not a transcription factor.
Mm-hmm.
But it interacts with transcription factors,
Mm-hmm.
the two activators.
Yeah. So it's a protein in the nucleus. It is regulating transcription, the turning on and off of genes. And it's not binding DNA directly like a transcription factor, but it's part of a complex of stuff that ends up regulating gene expression.
That's correct. Yeah. So that's what period protein is doing.
Mm-hmm. Mm-hmm. And so, obviously the world has a rhythm, right? We go around the sun, we have this periodicity to the physical environment. Animals clearly have behavioral rhythms they naturally go into, right? We tend to sleep at night, we tend to be awake during the day. People often eat at similar times of day, et cetera, et cetera. So we have this direct experience of all these rhythms. We see it in other animals. It sort of makes sense phenomenologically. You then told us people discovered, okay, there's this part of the hypothalamus, the suprachiasmatic nucleus. The neurons seem to fire in this periodic fashion. And then eventually technology improves and people like you and others start to figure out, okay, there's actually these genes encoding proteins that have a periodicity to them. They seem to be coming on and off at different times of day in this periodic fashion. Can you kind of walk us through the saga, an abridged version of the saga, and sort of talk about the dynamics of what's happening in the nucleus on the 24-hour cycle?
Yeah. So in the case of the period protein, the key was really being able to visualize the actual protein itself rather than the RNA.
Mm-hmm.
And again, these were just technical hurdles back then. And so it wasn't until the very late 80s that antibodies were made against the period protein that were reliable. And once that was done, they could see that the protein actually had a rhythm in the brain. And it tended to be enriched in the nucleus. So those were very big clues back then to sort of change sort of the direction for what the period protein function might be.
Mm-hmm.
Wasn't a cell surface protein, wasn't a glycoprotein.
Mm-hmm. So the protein itself, which regulates transcription factor, it's not technically, it regulates transcription, it's not technically a transcription factor, but it regulates gene expression. It seemed to accumulate and then go away in this kind of 24-hour cycle, this very rhythmic pattern. And, to anticipate, some of this, or to start to anticipate where we're going and connect this to what we were saying earlier, one thing that might be good for people to think about here is all of this stuff that we're talking about takes time, right? The protein has to be synthesized. It has to accumulate, then it has to do what it does. And there's kind of like this lag time, this accumulation time. And this starts to get you thinking about like why things exist on the time scales they do. Can you kind of give us a sense for like, what is our understanding today on the core transcription translation dynamics for how this molecular clock system actually works in total?
Yeah, so I would say the beginning now is um, two sets of genes actually activate the period gene. Um, those are transcription factors or activators, and the first one was discovered by my lab in 1997. It's called clock, as I mentioned before, uh, and it's a BHLH pass AB transcription factor. It means it has a DNA binding motif that was predicted when we cloned the gene. And it had other features that are characteristic of transcriptional activators. So the C terminus is glutamine rich, which is an activation domain for many transcription factors. So when we cloned the gene, it was pretty obvious what it might do.
Mm-hmm,
Uh,
mm-hmm.
Now, it turned out that it works with another transcription factor as a heterodimer. Okay, so the two proteins form a complex. That partner is called BMEL. And we discovered that about a year later with a colleague of mine, Chuck Weitz at Harvard, where we screened for proteins that interacted with clock. And we found that partner protein. And then we found that that complex actually bound to the original fly regulatory regions of the period gene.
Mm.
Because the complex binds to a specific sequence. It's called the E-box. And we knew that the period gene had an E-box. And so we actually showed that clock and BMAL from uh, mouse and hamster could activate the fly gene.
Hmm. So it all came together,
Yeah.
literally.
And then that same year, the period gene in mammals was finally cloned. It turned out to be extremely difficult to clone the period gene in mouse and human. And that's because at the DNA sequence level, it's highly divergent. You can barely recognize it at the DNA sequence level. But if you translate it into protein, then it's clear.
Mm.
It's a related protein. Um, And so between 84 and 97, when the mouse and human period genes were finally cloned, everybody tried to
Yeah, yeah.
go from the fly gene to the mouse or human
You
gene.
just look in the mammalian genome for something that looks like the DNA sequence in the fly, but it wasn't there, and that's because just of the details of the difference between the sequence at the DNA level between the protein level. It wasn't apparent in the DNA that you were actually
That's
looking at it.
that's correct. And so the human version of the period gene was cloned purely by accident. There was a genome center that was just sequencing full length cDNA clones on on a particular human chromosome,
Mm-hmm.
17, and one of those turned out to be the period transcript. And so when they translated that uh, mRNA sequence into protein, then it became clear that was period.
Mm-hmm.
You could see the sequence similarity with the period protein. The other way it was cloned was finally there is one region of the protein that has some conservation, and a lab in Japan just brute forced this region and made every possible combination of uh, amplifications of that domain, and they finally succeeded. And that's how they found it.
Mm-hmm. And so how does the periodic nature of this molecular system actually arise? I'm cheating a little bit because I kind of know some of the
Yeah.
answers here. Yeah.
Okay. So I would say it first starts with clock and BMEL. They bind to regulatory regions of the period gene, and then subsequently in flies, another gene was discovered called the timeless gene from Mike Young's lab.
Mm-hmm.
Um, and in the case of mammals, another set of genes called the cryptochrome genes were discovered. So clock and BML activate all of those genes uh, during the daytime. The transcripts accumulate, then the proteins are translated, and then they interact with each other. So in the case of flies, period and timeless interact with each other. In the case of mammals, period and cryptochrome
Hmm.
interact with each other. They form complexes with other proteins, and then they go back into the nucleus in the evening, and then they physically interact with clock and BMAL, their activators, and repress their transcription.
Hmm.
And
So
so that's a
yeah,
feedback loop that takes 24 hours to complete.
yeah. So certain genes get turned on by certain proteins. It takes time for these genes to go from transcription, making the mRNA, to translation, making the proteins that are encoded by that mRNA. Those things accumulate. And basically, when you just walk through the whole logic here, you end up with this negative feedback circuit where proteins accumulate that turn on some genes, then more proteins accumulate that end up repressing some of the other genes, and then you kind of just it all works out in terms of the timing and how long these things take, that you get this 24-hour periodicity in gene expression
Mhm.
changes.
That's right. Yeah, what's unusual is that the time constant is pretty long, even
Mm-hmm.
for transcription. So,
Mm.
transcription can occur in on the order of minutes to hours typically, but this
Mm-hmm.
is many hours.
Yeah. And so these genes are being expressed in this rhythm. There's the core genes encoding some of the proteins you talked about that I guess you would call them the core circadian regulators of the clock. But that's not all that's happening, right? Like there's many other types of genes that need to be expressed in periodic fashion. Can you talk a little bit about how the core clock ties into other systems, genes that
Mhm.
are going to be involved in the regulation of, say, metabolic shifts that need to be happening on a 24-hour cycle and other things like that?
Yeah, sure. So one of the major ways the core clock signals to the rest of the genome and the cell is that those master transcription factors, clock and BMAL, actually regulate thousands of other genes
Hmm.
in the cell uh, by directly activating them and turning them on in a similar fashion to, what we describe for the core circadian mechanism. Okay, so the major output is transcriptional, and literally thousands of genes are regulated in each tissue or cell type.
So the genes that are involved in this core clock mechanism, where you have this pattern of activation and repression of those core components that gives you what we call the clock. Some of those proteins are activating other genes, and it's not just a few other genes; it's a substantial proportion of the whole genome.
Yeah, that's correct.
Mm-hmm. And
And then some of those are actually also transcription factors.
yeah.
And then they activate another cascade
Right.
stream of
So
themselves.
yeah, so all day, every day, throughout our lives, throughout development, there are these exquisite and complicated loops of activation and repression happening, and they result in patterns of gene expression and patterns of protein accumulation that are ultimately going to dictate what the cell can and cannot do.
That's right. So about 15 years ago, when the technology became available to identify where these core proteins were binding in the genome, so that's through a technique called chromatin immunoprecipitation, followed by next-gen sequencing. So we could take clock and BMAL. We had good antibodies that we made against them. And then they bind to sites in the genome, in chromatin. You can shear the DNA into short fragments, and then with the antibody, precipitate those clock uh, BMAL bound DNA fragments. Okay. Then you purify those and you sequence them. And that allows you to then um, infer where the protein was binding in the genome through this method called ChIP-seq. Okay. So um, when we did that, for example, in the mouse liver, that's where I gave you that number 3,000. There are at least 3,000 genes that are directly regulated by clock B-mel uh, that are being turned on and off every day. There are actually even more genes where we didn't see obvious rhythms, but the binding was even more extensive than just 3,000 genes. It was more
Mhm.
like half the genome. Uh, so when we did that, um, that allowed us to then say, what kind of genes are those 3,000 genes that are being bound
Right, right. Is there
by
some kind of
BMAP?
pattern there? Yeah.
Right. So-called gene ontology.
Hmm.
And so when we do that, the top category is metabolism.
Mm.
That's kind of a general
Hmm.
category.
And when you say category, we are, this is we're humans imposing sort of these categories, giving labels to the different types of genes. Can you just give us some examples of what are the major gene categories that one would talk about and that were candidates maybe that you were thinking about at the time, just to give people some comparison as to like, okay, it's a lot of metabolic genes as opposed
Mhm.
to what other types of genes.
Yeah, so say in neuroscience, there would be genes involved in synaptic transmission, neural
Hmm.
signaling,
Hmm.
neurodevelopmental disorders, disease genes associated with various neurological disorders, psychiatric disorders. Those would be uh, under the broad category of neural genes.
Yeah, yeah.
Um, but then there are many categories having to do with cell biology, cell division, uh, and aspects of just maintaining the cell,
Hmm.
uh, genome stability, mitochondrial function. There are all sorts of different cellular functions uh, that come up as categories. Immune function is a huge
Yeah, yeah.
categories, for example.
Hmm. And so many of these things are going to be regulated by these genes, but it's not an even distribution. There was like this kind of disproportionate regulation that you saw of what you think of as metabolism regulating genes.
That's right. And so the way we visualize that is um, one of the sites that does this ontology has a metabolic chart of all pathways in the cell. And so we could then just label the genes in that chart. And what we found is the entire chart was covered, meaning every fundamental metabolic pathway in the cell was being directly controlled by the circadian clock.
And
And
just to
so
give
that
people,
was very eye-opening for
yeah,
us.
and give people a little bit more of a concrete sense, when you say metabolic pathways and metabolic genes, you mean genes that encode things like, say, proteins involved in generating ATP or that are part of the electron transport chain, like stuff that's to do with turning food, basically into energy.
That's correct. So there's the carbohydrate Krebs cycle, which is really uh, for generating ATP, but then there's lipid synthesis,
Hmm. Hmm.
various pathways for synthesizing other components, nucleotides for DNA and RNA, all those kinds of pathways.
And so, was that surprising when that was first coming out?
I think what was surprising to us is how pervasive uh, it was, and that the entire metabolic chart was covered. That was, we knew it, we had hints that the clock was involved in diabetes
Hmm. Hmm.
and obesity and regulation of lipid. Uh, that actually came from our original mutant mouse that we used to find the clock gene. That mouse, as it aged, became obese.
Yeah, yeah.
And when we studied it, it turned out it was diabetic.
Yeah.
had high cholesterol, high lipids.
Yeah, yeah. Well, this is perfect because I actually wanted to get here soon. So, one way that scientists like you in molecular biology broadly defined, one way that you go about figuring out what things do is, you can break stuff and see what goes wrong. You can delete a gene or mess with the gene, overexpress a gene, and so forth. And it sounds like what you're saying is some of the original mutants and some of these core clock genes, the animals that had that mutation, they didn't just have a weird circadian cycle. It wasn't like they were staying up late or had a compressed rhythm. They didn't only have those types of problems. They also had other problems like obesity and diabetes type stuff and all of that. Is that right?
Oh, yeah, exactly.
Yeah.
And in fact, they have shorter lifespan also.
Hmm. Hmm.
Um, so there are many morbid effects of having the clock genes
Hmm.
dysfunctional.
And so I'm just going to go with obesity, but we can go wherever we want with this. The animals that have these clock gene mutations that become obese over time in life, are they simply eating more, or are they getting obese despite the fact that they're not eating more, and it has something to truly do with the timing of feeding, say?
Yeah, so I would say on average, they're not eating more. Um, they're somehow metabolizing or processing those nutrients in a different way.
Mm-hmm.
And so they're, in the case of the clock mutants, they're storing more fat than a normal mouse. Uh, but they're not really eating that much more. That you can't account for that based purely on their eating behavior.
Mm-hmm. Mm-hmm. And so, what does this start to tell us about circadian biology and timing and metabolic health and fuel processing?
Yeah, so, as I said before, we are going through an energy cycle every day, uh, largely driven by our behavior.
Mm-hmm.
But what that means is our entire metabolism is tuned to process food while we're awake and eating, and not processing it when we're supposed to be sleeping and resting. Okay. And so that involves a whole mobilization of all these different genes and proteins they're involved in metabolic processes. So the majority of them have a circadian rhythm. They're cycling every day. Okay. And so it turns out that even though our feeding schedule is pretty regular under normal circumstances, uh, if we were to eat at the wrong time, there's still other nutrient signals that are used by our organs. Okay, so they can respond acutely to nutrient signals also.
Mm-hmm. Mm-hmm.
Um, and one of those, it turns out those signals actually can synchronize the clocks in our body. The clock in the SCN is resistant to that. It doesn't really pay attention to when you eat or anything.
Mm-hmm.
It's locked on to the light dark cycle. But for example, your liver and many digestive tissues, even your fat, are more strongly synchronized by the time of eating.
Hmm. I guess
And
that makes some sense because, right, your liver, a big part of what your liver does is, it can store glycogen
Mhm.
if you're eating sufficient amounts of certain things. So it makes sense that an organ like that would respond to the timing of feeding. It makes sense that the SCN and the hypothalamus, which is hooked right into the retina, would really be paying attention to external light. And this gets us to the idea, I think, that there can actually be sort of multiple clocks in different tissues, and they might be active or inactive out of phase with each other, but there are mechanisms in the body that start to align these things depending on whether you eat at one time of day or another, relative to when you sleep, say.
Right. Yeah. So, normally we're not changing time zones
Yeah.
or doing shift work. And so, under normal circumstances, the our brain clock is really controlling our behavior and sleep-wake cycle, and then that drives our feeding behavior, which in turn keeps metabolism in sync with our brain.
Hmm.
But if you were to artificially change when you eat and say eat only at night when you're supposed to be sleeping, uh, all your metabolic tissues actually are going to flip over and follow your eating time, and you will literally be uh, desynchronized. Your brain will be following the sun,
Hmm.
but your liver will be following your feeding time. And so we call that internal desynchronization. Uh, and it turns out that being internally desynchronized is actually not optimal.
Hmm.
Um,
By
there are
what
other
that
ways of demonstrating.
Yeah, but that makes intuitive sense. But by what measures
Yeah.
can we see that directly?
Yeah. So in human studies, there are very nice controlled studies where they can um, desynchronize uh, sleep time, okay, from your internal clock, okay, and under and they do this artificially by putting the the test subjects on either a 20-hour day or a 28-hour day
Hmm.
for sleep. So you can, your sleep can follow that extreme period, but it turns out that your main clock can't go to 20 or 28 easily. And so it sort of maintains a 24-hour rhythm. And so what happens is your sleep wake cycle is scanning across. Okay, if it's 20 hours versus 24.
Right, right,
Okay.
right.
And so at some points you'll be in sync, but at other times you'll be out of sync,
Yep.
right?
So if you do this day by day by day, you'll have different days where things are in sync or out of sync to some different
That's
degree.
right. Yeah. And so what they found is, glucose regulation is impaired when you're out of sync. And also insulin levels are impaired. So a number of different endocrine measures
Hmm.
um, can be impaired. Those were the first ones discovered, but others have been found subsequently. So just sort of that subtle internal desynchronization already is pushing you to a pre-diabetic state, it turns out.
I see. So basically, what this implies is that if you have two people and everything is completely identical about them, except the timing of these things, this will lead to different metabolic health outcomes. They could be the same body weight, they could be genetically identical, they could eat the same number of calories, the same diet in terms of what nutrients and calories they're actually consuming. But if one of them is eating at a different time than the other, you would
Mhm.
expect to see, based on what you just told us, that all of these different parameters, or many of them, are actually going to be different, and one person will actually end up healthier than the other over time if one of them is persisting in this more desynchronized state.
That's correct. And so, it's hard to study long-term experiments in humans are these conditions, but we actually done this experiment in mice
Hmm.
and looked at longevity, how long they live under these conditions. And so the experiment's a little bit more complicated than just eating at the wrong time because we had different comparisons. We have mice that eat at the correct time, which is at night for them, and
Yep.
mice that eat at the wrong time, which is in the daytime when they're supposed to be sleeping. But then we also had a group that were fed continuously.
How does that
Um,
how did that work operationally?
yeah, so these mice are under caloric restriction,
Hmm.
okay, because we're going into a different field here. Caloric restriction is really the most effective intervention for extending lifespan in model organisms. So, for example, in a mouse, if you reduce calories by 30%,
Hmm.
on average, the mouse will live 30% longer. It's a huge effect.
Mm-hmm.
Um, so under those conditions, the mice are hungry because they're 30% deprived
Yep.
on average. And so they eat as soon as we feed them, no matter what the pattern is.
I see. So you can just kind of plop food in there, and in that restricted state, they're going to wake up and eat it, even if
That's
it
right.
means waking up over and over and over again.
Right. So the group that was fed, I wouldn't call it continuously, but spread out over the day.
Even distribution of feeding time throughout the
That's
day.
correct. Yeah, that's a better way of saying it. Those mice eat exactly when we gave them the food.
Mm-hmm, mm-hmm, mm-hmm.
Okay, throughout like that. Um, and when we looked at lifespan, I think the biggest surprise was that group of mice that had spread out feeding, they still lived longer. They lived 10% longer, but they did not live 30%
Yeah.
longer.
So
Okay.
that's yeah, and that's a big difference.
Right.
Yeah.
And then the mice that ate at the wrong time, but they have a fasting interval of at least 12 hours, those mice live 20% longer,
Mm-hmm.
but still less than that 30%. Okay. And then finally, the mice that eat at the right time, which is at night, they live 35% longer.
So if the caloric restriction is restricted to one window of time and it's constant from a circadian perspective at the same time every day, that's where you see the biggest effect.
That's right.
Mm-hmm.
Yeah. And so, and the only difference in this experiment is the pattern of food intake.
Mm-hmm.
There's no difference in the amount of food per day because we record
Mm-hmm.
all of the eating behavior. Uh, and if you compare the ones that eat in the day versus the night, the wrong time versus the right time,
Mm-hmm.
the pattern is identical.
Yeah, yeah.
The only difference was
The timing.
the 12 hours of, the phase.
Yeah, yeah. The
And that
phase of
made
the
a
cycle.
15% difference in
Wow.
lifespan.
Yeah, so that tells you this is a big effect. We don't necessarily need to get into all the details here, but at a high level, is the way to think about this that, okay, obviously in a natural situation, an animal is going to eat when it's awake. And at some point, we'll stop eating and it will become behaviorally quiescent or inactive and it'll go to sleep. And, it's going to have these general kind of patterns in its behavior. And when it's asleep, it's obviously not eating and it's going to be processing or not processing things like food in, the wake versus sleep state. So when you're asleep, it's been a while since you've eaten. Your metabolism is not geared towards dealing with food and ingestion and nutrient extraction because you're asleep and you're not eating. And so that leaves sort of more energy available to do other things like, I don't know, what we might consider housekeeping, whatever is happening during sleep to sort of rejuvenate, the brain. There's stuff that can't happen while you're eating because all of that stuff takes energy and there's only so much energy to go around at any one time. And that's why the phase of the cycle is so important. Is that how you start to think about this stuff?
Yeah, I think the other reason the phase is so important is the mice eating in the daytime are eating at the wrong time metabolically.
Mm-hmm.
Our entire gene program
Yeah.
is designed to process feeding during the active phase,
Yeah. Yeah.
not the
So
rest phase.
all of the proteins and machinery that would be turning that food into molecular energy and doing all of that type of stuff, they're not fully booted up and online in that situation. And so you can't as efficiently extract what you need from that food.
Right. Yeah.
And so food seems to be a very powerful way to regulate some of the rhythms in the body and the behavior of the animal. But there are other things that can regulate timing as well. I wanted to ask you, when we talk about circadian rhythms, obviously light usually takes center stage because it's such an obvious thing to entrain an animal to, when we talk about the suprachiasmatic nucleus and the pathway from the retina to the brain, right? You're often talking about light. I wanted to ask you something I've always wondered about the whole field of circadian rhythms and photobiology broadly defined. And that is, when we do all of these experiments, they're done in laboratory conditions. And
Mhm.
obviously, you're not in natural sunlight conditions. And so the pattern, the intensity, the wavelength composition, all of the things that have been done in most of the experiments that have come to us from laboratory experiments, they are going to differ from the sunlight that animals out in the wild are experiencing or that we experience every day when we go outside. At a high level, how do you think um, how do you think that has affected how the field has progressed and sort of where some of the blind spots might be in terms of the use of artificial lights in laboratory settings?
Yeah, so I mean, in the laboratory, it's actually very difficult to mimic natural conditions unless you have your animals exposed to sunlight.
Mm-hmm, mm-hmm,
First technical thing is sunlight is extremely bright, much brighter than we can really realistically achieve in the laboratory.
mm-hmm,
Um, and so that's one limit. And then we certainly don't mimic dawn and dusk very frequently. We just turn the lights on or off,
mm,
um, acutely. Um, and so that's a clear difference. And then, of course, the wavelength composition does change with time, as we all know. At sunset, becomes a redder, long wavelength. Um, so those factors have been studied by various labs over history, um, and you can see quantitative differences in how animals respond to natural lights versus what we typically use, just
mm-hmm,
on-off light dark cycles. Um, but as far as you know, the majority of the effects of synchronizing the clock, those are well mimicked by laboratory cycles. Um, we would say you don't have to, have a completely naturalistic kind of light cycle, um, unless you're studying a really particular question
mm-hmm,
about the timing of when a rodent might leave the burrow or something like that.
mm-hmm,
Um, So yeah, so I don't want to downplay it too much, but I think that, when we're studying entrainment or synchronization of the clock, uh, our simple light cycles in the laboratory do mimic a lot of that.
mm-hmm,
Now, was missed in the laboratory historically is that the photoreceptors that are mediating entrainment, were really only identified uh, in this century, in 2002. So we've known for, over 100 years that we have two major photoreceptors, rods and cones, for night vision and color vision. Uh, but uh, it was really a surprise in the late 90s that if we looked at mice that had a mutation where all the rods degenerated, it's called the RD mutant. Mice don't have many cones, so all the rods degenerated, and most of the cones went with them. Those mice still synchronize normally to light dark cycles.
mm-hmm. Interesting. So this is a nocturnal species. They tend to be active at night. They've basically part of what you said is mice don't have as good a color vision as us, but they have better night vision than us based on how they're
Right.
set up to behave. You can create mice where the rods degenerate. So that's a big deal for a mouse that needs to be awake and active at night. And
Yeah,
even in that condition, the animal still synchronized to external light pretty much normally.
that's correct.
And what does that say about sort of the sensations, the light sensitivity side of this? It sounds like the core central clock, it's entraining to light. It really cares about light. You can't, it's not
Hmm.
going to entrain to food like the liver stuff does, but it sounds like it doesn't need that much light in order to do its entrainment.
That's correct. So, our visual system our night vision is exquisitely sensitive to light.
Hmm.
So our rod photoreceptors can detect a single photon, in classic
Yeah.
experiments.
Which, I mean, just to say it out loud, like that's an almost absurd fact. Like the fact that we can detect one is almost unthinkably sensitive.
Right. Um, and, our cone photoreceptors for color vision are extremely fast. They
Mm.
operate on a very fast time scale because, we can see um, a baseball's, um, traveling to us if we're a batter or a tennis ball at 100 miles an hour. And so our color vision is actually very fast dynamically.
Hmm.
Now, the clock doesn't require that.
Right.
In fact, the circadian system tends to ignore very rapid signals. So, for example, a lightning strike or something like that is very fast and too short to influence the clock. So instead, what the clock does is it integrates light over longer time periods. So when I was a graduate student, I measured the action spectrum for resetting the clock
Hmm.
in a hamster, actually. And what we found is that the peak sensitivity was at 500 nanometers, which is green. Okay,
Mm.
that's similar to the rod pigment. But the threshold was way higher, almost 6 orders of magnitude higher than a rod photoreceptor. Okay. Um, and sort of just above or near the threshold for color vision or cone photoreceptors. Okay. But there was, in the mouse at the time, there's no photopigment known in a cone photoreceptor that matched 500 peak. Okay. So that was unusual. But the really unusual thing we found is that if we traded off intensity and duration
Hmm.
when we gave light pulses, uh, the hamster could integrate light for almost 45 minutes, extremely long time period. Uh, and again, we know that the integration time for a rod is measured on seconds, and for cones, maybe 100 milliseconds.
Hmm.
So those are way faster than what we're talking about. So anyway, in 2002, a new photopigment was discovered by a person named Iggy Provencio. Um, and it's a visual pigment that's related to rhodopsin and conopsins, but it was expressed in a different cell type in retinal ganglion cells. Those are the relay neurons in the retina that send information to the brain through
Hmm.
the optic nerve. So, what Iggy found is those cells express this novel opsin
Hmm.
that he named it melanopsin because he isolated it in frog melanocytes. And so that defined a third class of photoreceptor
Hmm. Hmm.
in around 2000, 2002,
Yeah. And what's
which is
different?
very recent in
Yeah.
science.
What's different about those cells, those photoreceptors, compared to the rods and cones?
Yeah, so there are two major differences. One is they have a very slow response,
Hmm.
measured in tens of seconds to minutes. And most photoreceptors, if you have continuous light stimulation, they'll respond transiently, but then, the response goes away, it decays.
Yeah, they adapt to it.
That's correct. So these melanopsin cells really don't have much adaptation. They can respond to signals over minutes, many minutes, very slow. Okay, so that's the first thing. And then the second is their transduction mechanism is actually different, fundamentally different than rhodopsin and conopsins. Um, so I don't know if you want to get into that, but
I don't know if we need to get into the mechanics of that, but at the end of the
right.
day, what do these properties enable the cell to do that the others cannot?
Yeah, so it turns out that these photoreceptors are really ideally tuned to the type of signal that the clock needs to respond to in the environment,
Yeah.
meaning um, light that's bright, high
Hmm.
threshold,
Yep.
but also is around for a while,
Yeah. Yeah. Yeah.
so daylight is around for many
Right. Right.
hours.
And
It's not
daylight's very bright, so these neurons sort of care about bright light. They don't care about softer light, and they don't need to care about faster signals. That's why they have this slower integration time, because they actually
that
don't want to care about faster signals like a lightning bolt, as you said.
exactly
I see.
right.
What is like that type of stuff? Like, right. So if you think about this intuitively, a lot of this should make sense for listeners, I hope, right? When we're looking around, our cones, our color vision cells, they need to be very fast because you need to see stuff happening quickly. Their circadian rhythms don't need to be as fast. They want to, the sun changes slowly and continuously throughout the day. It's very bright. There's sort of these melanopsin cells we were just talking about are tuned to that type of change. When we think about all of these things, these different time scales of the different photoreceptors, how they're tuned to different intensities of light. You mentioned that these melanopsin cells that are important for circadian regulation, they're high threshold cells. They need intense light to be affected strongly. How does this start to help us think about things like the artificial light environment that humans are
Hmm.
now engaged with? So, it's great that a lightning bolt, which is very transient but bright, doesn't throw off my whole rhythm, because if you live anywhere with lightning around, that that would be really annoying. But now we have things like cell phones and computers, which can be quite bright and can be non-transient. Some people will stare at them for a long time,
Hmm.
even when the sun is down. How does the timing and stuff you were just talking about connect with how modern artificial light is impacting our circadian biology?
Yeah, so modern artificial light has a huge impact on our circadian rhythms because we're artificially extending the day into the night with artificial lighting. Um, the other factor is the wavelength. So these new melanopsin cells tend to be sensitive to blue light
Hmm.
and not to red. Okay. And so uh, there's been a clear emphasis on the wavelength spectrum, how much blue light versus other wavelengths, longer wavelengths,
Yeah.
are effective in synchronizing our rhythms or in keeping us awake at night. And so, for example, you're probably familiar that your iPhone or iPad may have the ability to change the wavelength of the screen. It's called night shift.
Yeah.
It it makes the colors more reddish
Hmm.
in the evening. And the reason for that is those longer wavelengths are less effective in affecting your clock
Hmm.
uh, than bright blue light or white light.
Hmm.
And so, uh, and it is true. I mean, it does impact us.
Yeah. So
Uh,
that stuff definitely works. The night shift mode, being sensitive to the amount of blue light coming out of the screens at different times of day.
yeah, as long as the intensity is low.
Hmm. Yeah. Okay.
So if the intensity, was long wavelength but still bright, it would still reset your clock.
Yeah, yeah.
So
So you can't just wear blue light glasses. You can't just put on night shift on your computer. If it's bright light in your face in the evening when the sun is down and you should be gearing up for sleep, that will still throw things off.
that's correct. Yeah.
Okay, so intensity and color both matter.
Hmm. Yeah. Now, one thing that has been discovered, which is really quite amazing to me, is that uh, during development in both people and in mice, exposure to shorter wavelengths of light actually affect the development of the eye.
Hmm.
And when mice or people are not exposed to blue light, or natural sunlight when they're young, the eyeballs become more elongated.
Yeah, yeah. Physically, the whole eyeball.
Yeah. And when your eyeball's elongated, it makes you nearsighted.
Yeah,
And so, for example, in Korea, the um, proportion of individuals who are nearsighted is huge
yeah, yeah, yeah.
and has increased over many decades, even in the United States. And it's because we don't really spend that much time outside anymore.
Yeah. So it's funny, when you start to learn about all of the biology here, the photobiology, the circadian biology, all of the stuff related to what the eye does and how it works and how it develops. I always just kind of I think back to my childhood and like the stereotype of the mother who was like, "Don't sit too close to the TV, go outside and play," and
Mhm.
there's a lot of truth to that. And you're saying that literally just the type of exposure, the pattern of photon exposure to the eye can physically change the whole shape of how the eyeball develops in a young enough animal.
That's correct. Yeah, it's amazing.
Yeah. Wow.
Uh,
And you see that type of pattern a lot. And on the one level, you're like, wow, that's kind of like amazing and maybe even surprising. But then when I reflect on it, it actually makes a lot of sense. Like if you the pattern of chewing in the mouth, how mushy or hard the mouth is, will develop craniofacial development in a human. What you're
mm,
saying is the composition of the light environment affects the physical anatomic organization of the organ that senses that stimulus. And that's, I think, a general principle, right? The pattern of stimulation early in development will affect the physical anatomic development of the organ that senses that stimulus.
mhm. Yeah. Yeah, I hadn't thought about it in that more general term like that, but
Yeah, yeah. Well,
yeah.
with that in mind, another thing I wanted to ask you about is when obviously light sort of gets all the attention or a lot of the attention when we talk about this stuff, and it makes sense. There's good reason for that. But, we've already hinted that light's not the only game in town when it comes to circadian biology. We talked about how feeding can impact circadian rhythms and metabolic stuff. There's a whole world we could have even gone into deeper there. I want to ask you about how another aspect of the physical environment might tie into circadian regulation. I think you've done some work in this regard, but I'm honestly not sure how much. But how does something like air quality in terms of oxygen content start to tie into circadian regulation? And this gets into things like obviously altitude and geographic or environmental location. It gets into things like air quality, but also things like exercise, right? Because exercise is a way that our oxygen environment gets changed. What do we know about how circadian biology is impacted by oxygen levels, say?
Yeah, so I'd say there are two parts to that question. One has to do with oxygen, the other has to do with exercise, which we should come back to.
Mm-hmm.
Um, so as far as oxygen and oxygen sensing, we know that there's a transcription factor that is involved in sensing oxygen, and it's actually a relative of clock B-mal.
Hmm.
It's in the same superfamily of proteins. It's called HIF1 and HIF2,
Hmm.
or the oxygen sensing proteins. And interestingly, HIF1 and HIF2 can interact with B-mal, the partner of clock.
Hmm.
So there's actually a direct molecular interaction uh, between oxygen sensing and components of the core clock mechanism.
Huh.
Very
And, and and
interesting.
and what type of proteins are these?
These are transcription factors
Oh, they
again.
are also transcription
Yeah,
factors. Okay.
yeah, same family as clock B-mal.
Okay. Okay. And how do they actually sense the oxygen?
Uh, they, well, they're involved in uh, binding uh, heme, which can sense oxygen. Um, and so that's sort of the general way they sense oxygen.
So heme, like let's explain for what heme is for people. Heme, like hemoglobin. This is a
That's right.
yes. So this is something that's good at grabbing onto oxygen.
Right. Mhm.
So you've got these transcription. So oxygen can be directly sensed by transcription factors, proteins that go into the nucleus and regulate how genes are turned on and off. And not only that, but these oxygen sensing transcription factors can actually directly interact with the clock transcription factors that we were talking about before.
Right. They crosstalk.
Wow.
The
Why?
two pathways.
Yeah. So why? How do we think about that?
Yeah, so I think we don't really understand that relationship very much.
Mm-hmm, mm-hmm.
Um, Not that much work done except just show that there's crosstalk.
Yeah, yeah. So there's crosstalk. There's a deep connection here. We don't know
Mhm.
nearly as much about this part of this whole system, but what do we know about like how sleep and circadian rhythms behaviorally are affected by things like changes in oxygenation? This could be a place where we talk about um, exercise, maybe.
Mhm. Um, yeah, I'm trying to think. There isn't, I would say there's less work on this aspect. Um, there's some, getting back to health, there's some interesting anecdotal evidence that living at higher altitude um, confers some metabolic benefits
Yeah, I've heard this.
in particularly in glucose regulation.
Hmm.
Um, and so I don't know how that happens, but I think that there is clear evidence that there is some benefit of living at higher altitude. Now, is that because there's less redox stress? Because we live in 20% oxygen most of the time. And oxygen, of course, we need, but it also has a dark side, right?
Yeah, yeah.
So the process of metabolism generates uh, redox uh, molecules, which are destructive to cell
Mm-hmm.
proteins
As a
and to
natural,
cells.
yeah. So as a natural
Right.
consequence of our oxygen-based metabolism, we're going to produce some amount of these reactive things that end up breaking stuff. And so the intuition, it sounds like the intuition, but we don't know for sure. The intuition would be higher altitude might be have certain health benefits because it's got a lower oxygen environment, and therefore, on average, there's going to be basically less oxidative stress.
Mm-hmm.
Hmm.
Yeah, and there's some experimental evidence for that.
Mm-hmm, mm-hmm.
It's kind of interesting. Yeah.
Hmm.
But of
And
course, the downside is when you have lower oxygen, that stresses you in other ways.
yeah, 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 yeah. So yeah, it quickly gets complicated. But
Right.
do we know anything about how acute changes in oxygenation affect sleep wake cycles and things like that?
I'm sure somebody knows, but I don't know very
Yeah,
much
yeah.
about that.
Okay.
Yeah.
Um, I want to talk about as well. So we talked about this concept of internal desynchronization earlier. You have your central clock that's really paying attention and trained to the light environment. But then you can have other clocks in your body, like I'll just call metabolic clock in your liver, and it may or may not be synchronized with the central clock. So these things can be desynchronized. And you basically told us that when those things are desynchronized with each other in your body, that's not optimal from a health standpoint.
Mm-hmm.
You basically want all of these internal clocks to be synchronized as much as possible. I'm guessing that's probably a general rule or principle here. Another type of rhythm I want to ask you about. This is again an area where I'm not sure how much you've done personally, but you can let me know. When we think about learning and memory, and we think about, obviously you have to be awake and paying attention to learn about new things, but at the same time, there's probably some amount of memory consolidation and things like that happening during the sleep phase. What do we know about rhythms in learning and learnability? Are there circadian rhythms in terms of like, an animal being able to acquire and remember things more at certain times of day or parts of its rhythm?
Yeah. So where it's been looked at, you can show a clear diurnal difference in both memory formation and recall.
Mm-hmm.
So, for example, in mice, you can conduct context-bent fear conditioning different times a day.
Mm-hmm.
So, um, we did that like many decades ago. And you can actually see a quantitative difference with time of day. And so, um, other kinds of experiments have been conducted are to look at long-term potentiation at different times of day. And again, you can see a quantitative difference. You can have more effective LTP at one time of day versus others. So I think what that's telling us is that, synaptic transmission is not just the same all day. It's likely being modulated in a circadian fashion.
Mm-hmm.
And obviously, for consolidation of memories, there's decades of work in how sleep is essential for consolidation of memories in many different ways.
Mm-hmm. Mm-hmm. When we think about so in humans, there's a lot of variability in circadian like behaviors or things. People tend to like to do things at different times, oftentimes. So, people talk about being morning people or evening people, night owls, and you see that kind of variability where on average, people tend to either be sort of more active or feel more of them optimal, more of their optimal self earlier in the day versus later in the day, even after the sun goes down. To what extent do we observe that kind of variability in experimental animals? And have we studied it at all? Do we know why individuals in a population might tend towards one or the other in terms of being more active on the earlier or later part of the cycle?
Yeah, if you look at wild-derived animals,
Mm-hmm.
there's tends to be a much broader range in, preferred activity time, the onset. So you can document that. But even in inbred laboratory mice, there can be significant variation depending on
Hmm.
the mouse strain. And so, those are presumably all close to genetically identical individuals, but there are many other epigenetic factors that are probably causing variability in their circadian systems. So I would say humans have an extremely broad range of preferred wake up time, say.
Yeah.
Um, and we now know that that's largely genetic. Uh, Many genes contribute to the propensity to be either a morning person or an evening person. And the majority of people are evening people.
Really?
Yeah, I would say in the laboratory, only 20% of individuals are what we would call genetically morning people.
Hmm.
The other 80% have circadian. So when I say that, what I mean by that is that if we measure the circadian period of a person in the laboratory under
Yeah.
controlled conditions, 20% of those individuals have a period length that's less than 24 hours. 80% have a period length longer than 24
Mm-hmm.
hours.
Mm-hmm. That's the norm for it to be a little bit longer.
Yeah. And if your clock is longer than 24 hours, that pushes you towards being a later person.
Yeah.
Whereas if your clock is shorter, that pushes you toward being an earlier person.
Hmm. And is there any pattern to that? Or is it kind of like, can you predict ahead of time who in a population will be the earlier person versus the later people? Is there any rhyme or reason to it and anything that we can use to explain why we see that pattern?
Well, as I said, it's largely genetically
Yeah.
determined. So if you were to genotype the person, then you have a pretty good prediction on their tendency. But of course, our behavior can easily be overridden by
Right.
social
Right.
factors or your job, of
Yeah.
course.
Yeah. Yeah.
So,
Yeah. One of the particular things I wanted to ask you about is melatonin.
mhm.
And I'll just ask you to start with the basics, but obviously there is a public perception about melatonin because of how the product, the melatonin supplement product out there is marketed and how people use it. The perception that people have is that melatonin is basically sedative, that melatonin will make you sleep. And so I want to talk about what's true or not true about that. But let's just start with the basics here. What is melatonin, and what is it doing naturally, and what's its natural rhythm in the brain and the body?
Yeah, so melatonin is a hormone. It's made by your pineal gland, and it's mainly synthesized and released at night in the dark. So it requires two factors: time, meaning nighttime, but also darkness. Light exposure, if you're healthy, will suppress your melatonin. Um, and so, yeah, reading your iPad late at night, that'll also suppress your melatonin, for example, uh,
Mm-hmm.
and delay your sleep. So it's a hormone that we would say encodes darkness. When melatonin is high, it's signaling to the body that it's dark outside.
Mm-hmm. And is that true for both nocturnal and diurnal animals?
Yes, it is.
So
Yeah,
it's truly a darkness related molecule.
that's correct. Yeah. And so, it's been studied for many decades. It has a very beautiful circadian rhythm in most animals, except for laboratory mice. One reason we think that's the case in laboratory mice is melatonin is in many other animals, birds and mammals that have annual cycles.
Mm.
It's involved in determining how long the day length is. Um, and so we think that's one of its important functions in mammals. So in sheep, which are short day breeders, and in hamsters, which are long day breeders, where this has been experimentally tested, you can actually um, control whether their reproductive system is active by infusing melatonin in that have different nocturnal durations. So long melatonin means long night, short day. So that would be a positive signal for a sheep and the opposite for a hamster. Um, and so those are beautiful experiments, very difficult to do, that definitively show that the duration of melatonin encodes the opposite of photoperiod, the darkness period, and that signal can be used to control reproductive status in animals that are seasonal breeders.
Mm-hmm. Mm-hmm.
Now, mice are not, strong seasonal breeders, but I think because they were domesticated, the major selective force is breeding when you're trying to make a mouse strain
Mm-hmm. Yeah.
from the wild. And so my assumption is that melatonin was bred out of mice because it's generally inhibitory to reproductive function
Hmm.
in most mammals.
Melatonin, basically high melatonin is inhibitory to reproductive function.
Well, it's encoding long versus short days.
Right. And if that is an important factor in the reproduction
That's
cycle
right.
of an animal. Yeah, yeah. Okay.
Yeah. But
So
anyway, for some reason, most laboratory strains of mice are deficient in melatonin.
yeah, wow, interesting. Okay, so then it's probably been understudied to some extent because
Mhm.
not in those animals. So you said this is a darkness molecule. It's
Mhm.
meaning it's going to go higher in the brain at night, whether you're awake or asleep, i.e., whether you're a nocturnal or diurnal animal. So, to what extent is melatonin driving sleep versus helping to set and synchronize the actual circadian rhythm? I've heard people say things like, "Well, it's not driving sleep per se or making you sleepy. It's more about setting your rhythm and people will often say things like, well, a melatonin supplement can be good for, say, jet lag when you need to shift your rhythm, but it's not necessarily going to help you fall or stay asleep. What can you tell us around that stuff?
Yeah, so melatonin really should not be thought of as a sedative,
Hmm.
something that will make you sleepy, except at high doses, which are supraphysiological. Um, they will make you a little bit sleepy, depending on the individual. Um, and so uh, we would say the most effective, or what melatonin does best is to reset your clock. And it does that at low doses.
Hmm.
At very high doses, like 10 milligrams or something, it will make you sort of sleepy, but it's not a sleeping pill. And so it, I think it really shouldn't be used as a sleeping pill per se. It's more to keep your circadian rhythm regular. And the big problem in the United States is that melatonin is treated as a supplement. And so it's not regulated by the FDA, and you can buy it, almost anywhere.
Yeah. And you said earlier, this is a hormone.
Yeah, it is a hormone, and unfortunately, it's not regulated. In Europe and most of the rest of the world, it's regulated
Hmm.
as a medicine. Um, so there was a paper published a few years ago that sampled about 30 different melatonin supplement products in the US,
Yeah.
and the the actual melatonin content of those pills was all over the place.
Yeah.
Some had no melatonin, some had way high melatonin, and others unfortunately had
Hmm.
contaminants. So that's potentially very dangerous.
Yeah.
Um, and so I think, unfortunately, the most common supplements on the market that you'll see in your drugstore have very high doses. They'll be like 10 milligram
Hmm.
pills,
So
which is
yeah.
way
So
high.
you're calling 10 milligrams a high dose. And
That's
so
correct.
many of them are labeled and supposed to be 10 milligrams, and that is a high dose. But what you're saying is also, depending on the brand, depending on the bottle, the dose advertised might be way different than the dose actually in the supplement.
Yeah, that's true too. Yes. Hmm.
Wow. And this is a hormone. And so by definition, hormones, they tend to coordinate lots of different things. I would imagine that melatonin isn't just doing one thing related to sensing dark to just synchronize circadian rhythms. That's very important. I would imagine it's doing probably a bunch of other stuff too. Do we know anything else about the effects of melatonin that maybe people should be aware of?
Um, yeah, I mean, there are dozens of processes there have been reported to be affected by melatonin, but um, I think that let's just say that not everyone thinks that melatonin does everything that people say it does.
Yeah, yeah, yeah.
Um, I mean, it's an antioxidant, but to have that property, it has to be at very high doses. Um, so I'm not sure it's like a longevity supplement from that standpoint.
Hmm.
Um, I think the main issue is really the source of the melatonin. There are companies that are more reliable.
Hmm.
They actually will provide you the quality control for their product. Um, and so I think the way to find which ones those are is to look at clinical studies done in the United States by reputable labs. I can tell you who those might be and
Hmm.
see what product they used.
I see. Yeah, so there's studies out
So
there.
that's what I take. I
Yeah,
do take melatonin for
you
jet
do.
lag.
Oh, okay. So you
Yeah,
you take it as needed for jet lag purposes because it does actually have an effect on resetting that circadian rhythm.
yeah, and it's a personal thing. I think you just have to try it. So
Hmm.
I worked on melatonin as a graduate student.
Hmm.
Okay, so because of that, I never took it. because it is a hormone.
Hmm.
But I'd say maybe it seems like 15, 20 years ago, no, maybe only 15 years ago, I had to go to Europe on a trip
Yeah.
because I was here. So it was less than 15 years ago. And then I came back and then immediately had to fly to China
Mm.
the opposite way. So I decided, okay, I'm going to try melatonin. And it did help me reset.
Hmm. Hmm.
So for transatlantic flights, I do use melatonin.
Hmm.
Um, the other thing that I find, this is again just, personal, unreliable information, is that there's a sleep measure called WASO, wake after sleep onset.
Mm.
So that's, you wake up in the middle of the night after
Right.
you've gone to sleep, and then you have to go back to sleep.
Yep.
And some people, once they wake up, they can't go back to sleep.
Hmm.
So I actually find that melatonin helps me with WASO.
Okay. So when you take it when you're traveling to reset your rhythm, that also it has an effect there as well.
Yeah, I think so.
Hmm.
But again, this is just,
That's just your personal
anecdotal.
anecdotal experience.
That's right.
Yeah, yeah, yeah. Interesting. What so what can you just like give us at a high level, how do you try and regulate your relationship to light and especially artificial light versus natural sunlight, given all the things that you've learned over the years about circadian rhythms and sleep?
Yeah, so one thing that is pretty important is regularity. It's again what your parents and your grandparents probably would have told you. Uh, Regular wake up time and bedtime actually are good for maintaining stable circadian rhythms. Okay. You clearly need adequate amount of sleep too. So that's independent of a clock. That's really for your sleep system. But and the recommendation is for 7 to 9 hours of sleep. Um, and many people unfortunately can't get 7 hours of sleep.
Yeah,
So
yeah.
that is an issue, especially as you age. Um, so I find maybe melatonin helps me there too, a little bit.
Hmm. Hmm.
Um, I try not to take it constantly,
Yeah.
but
Well, I
because it's
wonder,
a
yeah.
hormone.
Um, I actually haven't verified this. I don't know if it's true, but it seems plausible. I've heard people say before that melatonin tends to go down or become dysregulated with age because the pineal gland can become calcified with age, and apparently, quite a high proportion of people have a partially or completely calcified, pineal gland as they get older. And so, I wonder if there's something there where melatonin function in the brain tends to go down over time, as many things do, because apparently the pineal gland is particularly prone to calcification.
Hmm. Yeah, so melatonin levels definitely go down. That's been documented pretty
Mm.
well with age.
Hmm.
Why is that? I don't know if it's calcification, but
Yeah, yeah, but
it's
it goes
it's
down.
pretty clear that the amplitude of melatonin goes down with
Yeah,
age.
yeah, yeah.
Yeah.
And on the subject of sort of the practicalities of how to regulate one's relationship to light and the timing and the time constant stuff that we were talking about earlier, how, so a lightning bolt won't disrupt your circadian rhythms, but staring at a screen filled with blue and green light, for potentially hours in the evening is definitely going to affect your circadian rhythms. When we think about timing and brightness and how long these things take, are there any kind of guidelines that you can give people in terms of, um, you know, roughly speaking, how long before bed should you do you personally try to aim to like not look at the screens and stuff like that? Is there a decent rule of thumb there that people can use as a guide?
Yeah, I mean, my, personally, for me, actually eating time is more important than
Mm.
light exposure because Most of the work that we've done recently has to do with time of eating and its influence on longevity, aging, and health. And so, you've probably heard of the fad diet, intermittent fasting,
Yep.
where people try to eat in an 8-hour window. Um, we use that on mice, but in a different kind of situation, looking at lifespan. And in those experiments I told you about before, about phase of eating, um, we found that, a 12-hour fasting interval really was beneficial in the mice, at least 12 hours. Maybe we don't really need to go to 16, but I take that back. We've shown that 16 is slightly better than 12 in mice in recent experiments.
Mm-hmm.
Um, but the main factor there is the phase, the time,
Yeah.
the eating window, not just the fasting duration. Okay. And so that's why I made the comment that uh, eating is probably a very important factor at night. Um, Ideally, you would, you know, finish eating 3 hours before you go to sleep.
Okay. So, yeah, just to give people a number, it's not
Right.
a magic number, but roughly speaking, on the order of 3 hours before going to sleep is probably something decent to aim for in terms of not falling into this internal desynchronization issue that
Mhm.
we discussed earlier. And then
That's right.
obviously waking up in the middle of the night and having a literal midnight snack would be, the worst thing you could do from that perspective.
Mhm.
So, on the order of, whether or not you're going to do true time restricted feeding, whether or not you're going to be, a diligent faster, whatever exactly that's going to mean for you, 3-ish hours before bed is probably good in order
Mhm.
to avoid that internal desynchronization that you were talking about between the central clock in the brain that's tuned into light and then the metabolic clocks that are in the liver and other organs.
Right.
Got
Yeah.
it.
And then, most people recommend that you're winding down your light exposure, say 30, 60 minutes before you want to go to sleep.
Yeah, yeah. Yeah, and that's probably a good minimum. I mean, from my personal experience, I have turning off the lights and turning on non-blue lights for that last 60 to 1 to 2 hours. That makes a huge noticeable difference for me. If I'm not exposed to blue light for at least an hour, that makes a noticeable difference for me.
Okay. So
Yeah.
that may be working through your melatonin.
Hmm. Interesting. And
Uh-huh.
so one last little thing I want to ask you about before we sign off. So we talked about the the timing and the time constants of some of the different cells in the light sensing pathways of the body. And we talked about the melanopsin cells, and they're built different. They're not so much for fast image forming. They're more for the circadian stuff that happens on a slower time scale. And that was good because, we use the example of the lightning bolt. It's bright, but it's brief. And so it's not going to necessarily disrupt your circadian rhythms. Let's say someone wakes up in the middle of the night and they have to use their phone, something, there's an emergency phone call or something, and they're exposed to that light. Is there like a rule there around like, is a few minutes too much time? Can you do that for a few seconds and still fall asleep? How much of that like brightish light exposure from something like a phone is going to be sufficient to then upset your ability to fall back asleep, say?
Yeah, I'd say it's much less your phone. It's more your room lights.
Okay.
If you flick on your room lights and they're bright, that could have an effect on your ability to go back to sleep.
Mhm. Even for
Again,
seconds of time?
I think it's going to be on the order of minutes.
Mm-hmm.
Um, but if possible, it's better just to keep the lights always dim, you
Yeah,
know,
yeah, yeah.
so as not to disturb your sleep that much.
Mm-hmm. Well, Joseph, I want to thank you for your time. You're one of the gurus in this field, and you were one of the people near the top of the list for who I was going to try and talk to on this general subject. We covered a lot of ground. Obviously, there's a lot we didn't cover. This is a huge area. And part of the reason is because this is so fundamental, right? We talked at the beginning about how, almost, a very large percentage of the genome is regulated in the circadian fashion, because when you think about it, that's the whole life of an animal. Of organisms in general, actually, you have to have these circadian rhythms at all levels. Is there anything that you want to leave people with, or anything you want to reiterate from what we talked about today?
Yeah, I think one of the ideas we're working on now is that the strength of your clock actually is a very important component to overall health. We already talked about examples of how dysfunctional clocks lead to metabolic defects, but on the other side, there's a positive side. So, for example, we've tested the idea that, well, let me take one step back. We talked about melatonin levels declining with age.
Mm-hmm.
We see that same trend across all tissues when we look at gene expression, where the amplitude of cycling genes goes down. And our interpretation is the clock is getting weaker as we get older. Okay. And so when you have a weak clock, that leads to metabolic problems, as we discussed before. So the idea we're working on is, what if we could boost the clock, make it stronger with age? Would that have health and longevity benefits? So we've just finished that experiment. We have done that genetically with a mouse that has extra copies of the clock gene. And we found two amazing results. One is they live longer. They live 16% longer. They eat actually more than their normal controls, but they weigh less.
Wow. So, the naive interpretation of that is somehow their metabolism is just better and more efficient.
Yeah, they're much more resilient metabolically.
Yeah.
And then when we challenge them with high-fat diet, they're actually resistant to weight gain.
So they don't gain weight as readily in response to the diets that cause normal mice to gain weight.
That's correct. So we think they're metabolically more resilient by making their circadian clock stronger, and they live longer.
Yeah. And remind me, how did you make it stronger? You're just giving them additional copies of some of these master genes?
Yeah, we have a transgenic mouse that we made that has 3 extra copies of the clock gene in it.
Wow. So 3 do we normally have just 1 copy?
Well, we normally have 2 copies.
Yeah. Well, yeah. Okay. Yeah. 2 copies. So 3. Okay. So 3 extra copies.
Yeah.
On top of the 2 that we normally have, because we have two were diploid. Does that work out?
Maybe it's 6 extra copies because it's 3.
Oh, okay. There we go. Yeah. Okay. Okay.
Oh, no, it's not. I hadn't thought about it this way. No, it's only 3 extra copies.
Okay. 3
plus
period, not 3
yeah.
pairs. Yeah.
Right.
Okay. But extra, you're more than doubling the horsepower there, I guess.
That's right.
Okay. And you get those amazing effects. Is that work published?
No, we're getting close to writing it.
Interesting. Yeah. Okay. Yeah, I will look for
excited.
that. Yeah. I was like, how did I miss that? Interesting. Okay. Well, if that's it, I think this is a great spot to leave it off. I'm looking forward to this new result coming out. Thank you again for your time, and I really appreciate it. Is there anything else you want to say?
No, my pleasure. Thank you for the invitation. It's been great talking.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.